Hi, I'm Anna Pickin from the School of Cancer Sciences. The title of my talk is Specifically Targeting an Off Molecule, a Novel Approach for the Treatment of Leukemia. In the past 40 years, cancer survival rates have more than doubled. This means that for the first time ever, as many people survive cancer as die from cancer. But more work needs to be done, as in the last year, in the UK alone, over 160,000 people died from this disease. So I work on leukemia, which is a type of blood cancer. In order to understand how leukemia occurs, it's important to consider how our blood normally develops. So in our blood, there are many different cell types. We have white blood cells, which fight infection, platelets, which help our blood to clot in response to injury, and we have red blood cells, which carry oxygen all over our bodies. And despite their vastly different functions, quite remarkably, all of these different cells began life as the same cell type, the blood stem cell. So these blood stem cells live in our bone marrow, and they can choose to renew themselves and stay a stem cell, or they can choose to mature, and they can mature down the white cell route or the red cell route, and so on. However, regardless of the route taken, this maturation process is always very gradual, with several intermediate stages of maturity being met before fully functioning blood cells are made. Now in leukemia, these stem cells begin their journey, but they are stopped and blocked at an early developmental stage. And the prolonged duration of this ordinarily quite short-lived stage increases the chance that these immature, non-functioning cells are going to obtain a cancer-causing mutation. Now, upon mutation, these immature, non-functioning cells grow far too much and take the place of the normal, healthy blood cells. And the consequences are so devastating for that person because they now essentially do not have enough functioning blood to survive. Now, thanks to work in my lab, as well as several others, we actually managed to work out why these stem cells get blocked in that early stage in the first place and are therefore predisposed to cancer. So an off molecule within the cells, which normally binds the DNA and turns on maturation instructions, is abnormal, so it now binds the DNA and turns these instructions off. So I grew these leukemia cells in a petri dish, and using a specific laboratory technique, I was able to remove this off molecule. And as a result, these immature cancerous cells started to mature again and look and act more like a normal blood cell, suggesting that moving this off molecule reverses this block in differentiation and triggers the cells to grow up and behave properly. This result was really encouraging, as it shows that targeting this off molecule could be a novel way of treating this type of leukemia. Now, the exact method which I use in the lab is not a feasible therapeutic route for a person, so efforts are now being made to design a specific drug compound which can target and stop this off molecule from working. I'm optimistic that eventually this work will lead to the development of a more specific and therefore more effective therapy for leukemia and ultimately save lives.